fine, as long as you're peeing it out. Okay, that's what an INO is, an intake and output. So you don't want to have a high salt intake and drink lots of fluid, because that's just going to retain everything and increase pressure, pulmonary tension, and that can increase, exacerbate problems with
So like if we were drinking like a lot of coffee, does the caffeine into that?
Caffeine itself is, well, caffeine is often called a diuretic. We'll see that in lab today. It doesn't appear to work directly on the nephrons, but on the blood vessels, causing increased pressure via the increasing blood pressure and therefore increasing urine output, rather than being one of the diuretics that works here. While we're on it, dehydration. I couldn't find any person that was dehydrated, so I put in this little marsupial. All right, so insufficient water intake or excessive water loss. And so what does alcohol do? Is alcohol an anti-diuretic?
Diuretic.
It's a diuretic, not directed again. It's kind of like caffeine. All right, alcohol blocks the release of ADH. And so ADH is working over here to open up aquaporins. So if you had elevated alcohol intake, then you would have fewer aquaporins in the collecting duct. Therefore, the dilute filtrate is going to be allowed to leave the body without being reabsorbed, whether or not your body needs that food or not. You're not going to have as many water channels as you need. So again, alcohol doesn't work directly on these structures, but works on the brain as far as inhibits the release of ADH. So insufficient water intake or excessive water loss. And again, this creates too high a concentration of sodium primarily, because that's our primary extracellular ion. And it can have CNS effects as well. In fact, sometimes the runners can tell you it's difficult to tell the difference between overhydration and dehydration. Okay? So these are the major electrolytes that are affected by water gain or water loss. Potassium is the most dangerous one. So that's why someone that has heart problems already, they would use potassium-sparing diuretics. All right, to watch those. But again, it's because, well, all of these, calcium, potassium, and sodium ions affect muscle and nerve. You have your heart muscle. You have your brain function. Nerve function to the heart. So all of these become affected. Notice I'm being pretty general. I'm going to stay general, okay? So this is not a nutrition class. It could become a renal physiology class and get really hairy really fast. In fact, I was telling the morning class, they didn't believe me, how lucky they were when I was showing them the slides of the ADH and the G proteins. I was talking the same, telling study that had a student at set state who gets migraine staying up all night to study. And they're also taking an upper division physiology class, all right? For biology majors, it has a 100, 130, 131 number, which is upper division. They're doing endocrine system this week. On Thursday last week, they talked about pituitary, no, Tuesday this week, they talked about pituitary, thyroid, parathyroid. And I said, well, have you done the physiology of the endocrine system already? They said, what do you mean? And I said, well, have you talked about receptors, G protein pathways? And they're like, no, we're not even getting into that. Like, well, this is upper division. That should be, knowing the names of the hormones should be something that you got, you know, in general biology or something. And so they said, well, our teacher dumbs down everything, and we get to lab, and our lab instructor kind of explains things. But then they explain them opposite. So the lab instructor will say, well, this is the way it really is, but answer it the other way because your other teacher is the one that writes the test. Wow, like, okay, I can learn the right way and the wrong way. But anyway, so you guys, I know that endocrine system was hard in learning those pathways, but that's what a lot of our pharmacology and neural control and everything is based on. So you're going to have a leg up on other courses in which those become important in understanding pathologies. Okay, Micturition we will do on next week, and I'm going to finish this now rather than come back after lecture. So let's look briefly at urine characteristics. We're going to be doing that for lab. Unfortunately, we don't do body fluids here, but other faculty and I are looking forward to the next semester too, where we're going to see streams of students walking back and forth between the restrooms carrying little cups of urine. Since we can't do that, so one of the, it's a fun lab that I've done with other classes, and Sac State does do body fluids, including blood. And students divide up into groups of 3 or 4, and they're asked not to go to the bathroom. They have a morning class and they get up in the morning. They have an afternoon class not to go to the bathroom after they get up until they come to go to the bathroom then, but not go again until they come to class. Figure out their kilograms of body weight and then drink a certain volume based on their kilograms of either cranberry juice or a high sucrose soda, distilled water. And a few times people have asked permission to do the alcohol. And I was like, okay, that's fine with me, you can't drink it in the classroom. So they were in the 1 o'clock lab, so they came in and did their, first peeing and getting rid of the bladder, the urine in the bladder, and then when they serve alcohol on campus at Sac State, and went and drank the prescribed alcohol, and then came back and did the rest of the labs. So they got to see the effects of that. But when you look at the urine samples, which are fake urine, by the way, I have a recipe for urine. I used to know which is apple juice. People would smell it, go, this is weird. When we look at volume, the range, remember I said the normal is 1,500 mils to 2 liters. And so less than that, it's going to be more concentrated. More than that, it's probably going to be more dilute. So when we look at the color test, we have these ranges of color. So in lab, when you're asked to describe color, you can say pale, straw color, amber, dark brown, whatever shade of yellow or brown. And that's usually due to the presence of the bilirubin breakdown products, the red blood cells, that have been added via the blood. Of course, the darker the color, the more particles are going to be present. Okay, the more concentrated it's considered to be. Get that. Turbidity refers to cloudiness. So if it's been in the bladder for a long period of time, between voiding, which is another word for micturition, which is another word for peeing. I can never remember it's number 1 or number 2, but cells tend to slough off the lining of the bladder wall. Okay, an infection with white blood cells and pus can make it cloudy as well. Precipitates of calcium if someone has kidney stones. So anything addition to the normal constituents can increase the cloudiness. Here you can't even see the graph paper through the light color. It's not really concentrated, but it's definitely cloudy. Odor. So some people, within 5 or 10 minutes after eating asparagus, if they go to the bathroom that quickly, it's already absorbed across the digestive tract wall. Not even time to break down the cellulose or anything like that. Get in the bloodstream and get filtered out and in the pee. Okay. Some people don't do this, but it's controversial whether it seems to be easy to test. I was looking it up for some students this morning. The question is whether they don't make it or they can't smell it. Why don't you just take someone in the bathroom and you say, you smell this? It's the chemicals methanyl. It wasn't a form of international nomenclature that I recognize. Obviously, urine has been exposed to the air, so baby diapers or animal urine in a rug gets a strong ammonia smell from the exposure to oxygen. Okay? But we have additional smells. So sometimes urine has a sweet smell. If someone's a diabetic. Is it the sugar in the urine that you're smelling? You might taste the sugar if you stuck your finger in it. But someone who's not able to use, you guys keep turning your nose up at that. Someone who's not able to have sufficient insulin to internalize glucose into the cell. That's the major fuel for our body's metabolic activity. So they start breaking down proteins and fats. And one of the products of breaking down fats are ketones. Okay? And that sweet smell is from the ketones. Okay, kind of like an acetone, nail polish type of smell is what occurs. We have one here that should have a really faint one. You may not smell it in your test tube, but if you smell it in the larger jars, I think number 2 should have an odor like that. pH, we already talked, the range is about 5 to 8. All right? And it's those buffers that keep it from being below 5. And so we have the pH indicators here, all the way from 0 to 14. So you can, sometimes they will be on the keto sticks, but they aren't on these. Specific gravity. Now I was going to set up an observation for specific gravity for our sample urines, but I didn't have enough to float my boat. So this is a basic form of a urinometer. We bought some for the marine biology classes and field classes to take them. But anybody know how specific gravity is determined? It's a ratio, like our FeV1 over FeVC. But what's it a ratio of? Notice the normal range for urine is 1.003. That would be a dilute urine. To 1.03 would be a more concentrated urine. What is making the specific gravity higher?
Water.
No. Remember I said more concentrated? So we need the particles. Urine with a higher specific gravity is going to have a higher osmolality. So it could be glucose, it could be salts. So you would expect the specific gravity of water from the Red Sea with a high salt concentration to be very high. So specific gravity is determined. The specific gravity of distilled water, double distilled water, is 1. So you take the mass of the water and divide by the volume. So 1 ml of water weighs 1 gram. Okay, so when mass is equal to volume, the specific gravity is 1. When we start adding solutes, like glucose or salt, we can dissolve quite a bit in there before it actually changes the volume. All right? So it becomes heavier. If you go to the bathroom and you have a very dark, concentrated urine, it sinks to the bottom of the toilet bowl. Okay? Whereas if it's very dilute and very pale, it easily distributes and diffuses, unless of course you have blue water and then you can't tell where it is. Unless there's green or something. But so specific gravity, the more dense the fluid is, the higher up this little ball will ride. Okay? If it's not very dense, then it's going to sink and be closer to the specific gravity of water. Okay? So think about floating on it's easier to float in salt water than it is in fresh water. And that's because of the higher osmolality, which contributes to a higher specific gravity. Okay? So if I were to have a picture of this on the lab exam next week, you should be able to recognize that this is a piece of equipment for testing specific gravity.
Oh, I looked up the compound in asparagus. It's called S-methylmethionine.
Okay, I have the meth part right. It has a couple of sulfide groups in it. So you would think they'd be able to tell whether people can smell it or people can actually make it. And did it say how the biology of how it's produced? Because the site I looked up, they said they still didn't know exactly.
The full article, they just gave me the abstract from an LA.
Okay, I think white asparagus is supposed to make smell more than green. I think white asparagus is weird anyway, but. All right, so on your quiz, there'll be one question off the bladder. So I wanted to go over that first. The rest of it is histology. Okay. Everything that you see here is retroperitoneal or down in the pelvis. So remember, our kidneys are retroperitoneal. The blood supply to the kidneys is retroperitoneal. The pelvis and ureters travel down the posterior abdominal wall, retro to the peritoneum. They then cross over the pelvis and down to the bladder. Now every time we look at the bladder, we see it round. It's a little joke. One of Gary sometimes cause low back pain. That's referring to. The bladder tends to be collapsed. So the first time I dissected a cadaver when I was a graduate student at the medical school, I thought my poor guy didn't have a bladder. I'm looking down in the pelvis and there's nothing there. But it's collapsed, and if this is my symphysis pubis or his symphysis pubis, instead of being a big bladder right there, it's very flat and it looks like it's just the membrane covering over the bone. Okay? So when it's empty, it is actually quite a flat structure. The ureters are the two tubes that form from the pelvis, and they come in at an angle. Here's the opening for the ureter. Ureteric orifice means the opening of the ureter. And it passes through the bladder wall at that angle, so as the bladder fills up, that tends to form a natural valve that closes off the ureter and doesn't allow the urine to back up into the ureter quite as easily. You guys already remember the muscle? We covered that with reproductive. Detrusor. Okay, dartos is the scrotum, but they're both smooth muscles. Do you have a question?
I may have just misheard it a moment ago. You may have answered it. On the quiz, the practice quiz that you have online, it identified the muscles of the ureters as detrusor muscles. Is that correct?
There's a circular and longitudinal and circular give, but I don't think they're called detrusor. The muscle of the bladder is the detrusor.
That's what I thought. I guess
Yeah.
it was just
So okay, I'll have to. On Tuesday, we'll look. Is anybody in here doing the Micturition reflex case study? The lady that can't keep her panties dry and wants to go to Burma and ride elephants.
Oh, awesome.
And then she's going through, okay. If no one's doing that case today, I'll go into it more. The Micturition reflex is very similar to our reproductive reflexes, the same pelvic splanchnic, sacrosplanchnic, pituitary nerve type of component. So, is going to the bathroom. Well, first let me finish the structure. So, detrusor muscle. The trigone is this triangular smooth area between the two ureter openings and the urethra. So we have two ureters and a single urethra, right? And then the rest of the lining of the bladder is thrown into folds to allow it to stretch and expand and collapse again. And those folds are known as rugae. We'll see that again. We saw it in the vagina. We have those folds of rugae in the vagina. And we'll see it again in the stomach. So the bladder is smooth muscle. Its contraction is under the control or stimulated by parasympathetic or sympathetic. Good, parasympathetic, which always throws people off because it's like you wet your pants when you're scared to death. Okay? We talked about that a little bit. The phenomenon is not completely understood, but it's thought to be related to the fact that an extreme amount of sympathetic activity causes constriction of blood vessels. The smooth muscles lose their blood supply and oxygen and relax. And that allows urine to flow out of the bladder. So the normal function of sympathetic is to inhibit contraction of the bladder wall to allow it to fill and increase constriction of the internal sphincter, which is also smooth muscle, to keep urine from leaking out. Okay? Then, as the bladder wall stretches, sensory receptors travel back to the spinal cord to trigger the activation of the parasympathetic nerve, which now is going to do the opposite. It's going to contract the bladder wall and relax the internal sphincter. Now, if that's all it took, we'd be carrying an extra pair of panties and trousers and underwear around with us all the time. Okay? Part of bladder training is the inhibition of the external urethral sphincter through the descending neurons from our frontal lobe, so that we don't go until we're actually in a proper place. All right? Which for me was my own bathroom at home until I was about 18. When I was in high school, I never, ever, ever used the bathrooms in the high school. And until I got home about 5 o'clock. We drove from Maryland to my grandparents' house. It was about a 9-hour drive. The rest of the family would stop at the restrooms at the gas station, and I make a beeline for the bathroom as soon as I got to Grandma's house. But it was only a problem when I went camping. And a friend of mine, I was teaching at college, and a friend of mine was teaching high school next door, and he was taking the class on a camping trip and needed a chaperone. I about 5 miles up onto the plateau in Tennessee, and it came to be dinner time and time to get ready for bed, and I went to find a nice little log for my toilet seat. And So I learned as a nurse that you can increase this sensory thing by putting your hands in warm water. When you're camping, that doesn't help. There's no warm water. Listening to waterfalls, so turning the spigot on. This is with women who have had children. They have a hard time going to the bathroom afterwards. You get a little ketchup bottle, you squeeze warm water over their perineal area, and put their hands in warm water in the sink, but none of that was available camping. So I gave up and tried again before I went to bed. And the next morning, went and brushed my teeth, and okay, I'll try to go to the bathroom again. And nothing worked. That voluntary inhibition was just way too strong. I don't know what would have happened, but about 3 o'clock that afternoon, a thunderstorm came up. And so we hiked back down and drove 2 hours home, and by 11 o'clock that night, I was finally in my own bathroom. But now, I've gotten used to the world. And I can actually go outside when I'm camping. So, but that has to do with those inhibitory effects, okay, and learned behavior once, a child gets potty trained. So we'll look at those more specific patterns on Monday. Okay, so we'll stop here. Give you some time. 35, this morning took about 35, 40 minutes. So I'll check with you at about 2:15 and see how